Okay, good afternoon. Uh, Today, we move to the next section, okay? I'm not so sure how many of you ever learn pharmacology, but don't worry. We try to teach you just only concept of the antibiotic, okay? Let's start from the word chemotherapy. Chemotherapy is the use of chemical for treatment of infectious disease. All right. Uh, Hang on. So you can see my slides properly, right?
Yes,
In my screen. Okay, it's come back. Let's continue. So in the old day, people use medicinal plant for treatment. Okay, until 1909. So Ulrich, German scientist who used chemical to kill bacteria, but not harm the host. So that is the first time we use chemical for treatment until, well, it's about 10, 20 years later, Sir Alexander Fleming found penicillin by the fungal Penicillium notatum. As you see the plate of Staph aureus, well, everywhere the fungi grow, around the colony of fungi, bacteria cannot grow. Okay, that's the first found of antibiotic. But later on, not just that day, but about 10, 20 years, so people just developed antibiotic to use, but penicillin was found in that year. I would say antibiotics are a cornerstone, very important for modern medicine because every time doctor trying to do something with patient, so antibiotic will take important role, including the wound infection. If you get wound and antibiotic help the wound clean and clear from infection. Pneumonia, there's another infection, blood infection, UTI. Do you know UTI, urinary tract infection? So you get infected in the urinary tract, since kidney, urinary bladder, and all tubes. Gonorrhea, sexually transmitted disease, complication delivery. That's mean well, when mother deliver children with a very complex, well, sometimes mother get infected with a virus, something like that. So bacteria, doctor need to supplement with antibiotic to stop the disease of mother, to save baby. Okay, injury, operation, transplantation, or even treatment of cancer. So in case of cancer, doctors still overuse antibiotic to prevent infection. That's important. So antibiotic is now being a key of the successfully in medicine. All right. So before we go detail of the antibacterial, let's know about the terminology. Chemotherapy. Chemotherapy just use chemical against the microbe, okay? Sometimes people call chemotherapy as antimicrobial drug because we jump from the state of chemical, pure chemical, to antibiotic. Antibiotic is the chemical, actually, the substance that produced from fungi or other bacteria, but action against microbe. But now the term antibiotic just point to bacteria. Okay, some chemical that affect with fungi, so now we call antifungal instead of antibiotic. The chemical that affect with virus, we call antiviral. So we now mean antibiotic is antibacterial. Okay. Once we use some drug or antibiotic to kill bacteria, bacteria also develop some weapon or some mechanism to fight back with the drug. That's called antibiotic resistance. Okay, antibiotic resistance refer to ability of bacteria. Well, now we talk about bacteria to resist, to not get the effect of antibiotic. This table just show the source of antibiotic from fungi and some bacteria. Okay, we get penicillin from fungi penicillium, something like that. We also get streptomycin from streptomyces, something like that. Bacillus polymyxa, we get polymyxin. This is in some example. Okay. So selective toxicity is the thing that we should concern. In this case, it means any drug that harm microbe, but not harm the host, but not harm to human. This is very important. Any antibiotic, when we develop antibiotic, we should think about the efficacy.
อาจารย์ค่ะ
ครับ
Sorry to interrupt นะคะ There's a student trying to join the class but she cannot นะคะ I think she sent a request นะคะ
Okay. So next time should come before, okay? So medical thing is very important. On time is important. If you're a doctor, you late for 5 minutes, patient may die. So this is the basic concern of the medical class, okay? So please, when I concentrate on teaching, that is another screen. Okay, back to the selective toxicity. We need to think about the efficacy and toxicity, right? The efficacy is ability that the drug can kill microbe. But toxicity is the effect of drug that happen on human. Sometimes you take, this is not about the allergy of drug, okay? But when you take some antibiotic, it might get toxic to your kidney. That's toxicity. So when we calculate the therapeutic dose, the amount of chemical that use and kill microbe, and amount of chemical that use and get toxic to human, we need to calculate the dose for toxic and dose for therapeutic. The toxic is supposed to be high, okay? Therapeutic dose supposed to be low. When you put the dose in the equation, the result is chemotherapeutic index. The index is higher is better. Toxic dose high, that means you need to eat a lot of drug, then you get toxic, not just few amount of them, okay? But therapeutic dose, you no need to add more, just few of them, and then disease kill. That's good. So the toxic dose and therapeutic dose supposed to be different. So we have big gap, okay? For bacteria, for antibacterial, lots of them are quite good. Toxic dose is quite different from therapeutic dose, but some antifungal, because fungi are also eukaryotic cells made from eukaryotic cell like us. So therapeutic dose may be quite close to toxic dose. That means if you take some more drug, it might get toxic, and that is a side effect of the drug. Okay. All antibiotic, they action in two modes. One is bacteriostatic. Bacteriostatic is not kill bacteria, but let bacteria stop growing. Okay, stop growing is mean stop dividing cells. All right, but not kill bacteria. Once you remove bacteriostatic drug, bacteria keep multiply and grow. Another type is bactericidal. Bactericidal is a drug that can kill bacteria, not stop. Even you remove the bactericidal, bacteria die. Okay, that's two things. Why we need to have two things? Generally, we in normal patient, just like you guys healthy, and one day you probably coincidentally get infected. So doctor might give you a bacteriostatic drug to let bacteria not grow more than this, and waiting until your immune response clearance of all bacteria from your body. Okay, that's the perfect one. So bacteriostatic and bactericidal are both effective in normal patient, but immunocompromised patient cannot develop immune response, and without no immune response, even innate immunity, patient cannot tolerate the effect of bacterial infection. So bactericidal is more effective than bacteriostatic. The question is, why normal patient, why not use bactericidal, get rid of disease or bacterial infection in normal patient? Why we need to waiting for immune response? Do you know why?
Maybe it involves with the microbiome.
Involved with what?
Maybe by not killing or involved bacteria, you can still retain the microbiome and induce an immune response.
That's why we need to let immune get rid of or clearance bacteria instead of bactericidal.
To prevent the bacterial resistance in the future.
How?
Like
to develop immune memory.
Yes, we need memory. If you clearance bacteria too quick, so immune not develop until you get memory cells of memory B, memory T, so then you might get infection over and over and over. And some disease, when you get second time infection, the disease may respond quite toxic more than the first time. It probably happened like that. Okay, when we know the drug can have two modes: stop bacteria, kill bacteria. Okay, and what type of bacteria they stop or kill? That refer to spectrum of antibacterial, antimicrobial activity. Okay, we have the term broad and narrow spectrum. Both of these terms is quite subjective, but the broad spectrum is mean this antibiotic can affect to most of bacteria, both including gram-positive. So many type of gram-positive get affected, and also gram-negative bacteria. For example, tetracycline. Tetracycline kill lots of bacteria when you take it. Okay, the narrow spectrum antibiotic, we point to some group of bacteria, but not all. Okay. For example, either gram-positive, some antibiotics, for example, penicillin. Penicillin are active very well with gram-positive bacteria, okay, but not gram-negative. Or isoniazid. Isoniazid effect on mycobacteria than the other bacteria, okay. That's the spectrum. Which one you think we prefer? Which one is better? Broad or narrow?
Broad spectrum,
Because
it can kill many type of species of bacteria.
right when you right is mean it can kill many species of bacteria. But when you take broad spectrum, the good bacteria in your body also die. We need narrow spectrum, kill particular bacteria that we don't want. But normally when patient go to see doctor, you guys get infected, go to see doctor. Doctor need to observe, diagnose what kind of disease, what type of bacteria that infect you. In case that doctor not so sure, in some case, he probably think maybe 3 or 4 possibility. When doctor prognose, he think in a broad sense, maybe 3 or 4 possibility, and all 3 or 4 are gram negative. Then he get broad spectrum gram negative, and then take your blood sample, go to send to the lab, or maybe next week come to see doctor again. Once the lab report come out, he know or it is the causative agent, then he change the medicine. Then this is narrow spectrum to kill that bacteria. Okay, this is the idea. Okay, this picture show the spectrum of antimicrobial. You see, we classify bacteria to be gram-positive cocci, gram-negative bacilli, negative cocci, and aerobic or very unusual bacteria. No need to remember, but I want you to see penicillin G. Penicillin G work well with streptococcus. That is narrow spectrum. But a 4th generation cephalosporin can work well with MSSA, gram-post cocci, gram-nec bacilli, gram-nec cocci, quite a lot. So this can kill most of bacteria, okay? That's as an example. When you take antibiotic, or anyone take antibiotic, in your body has normal flora, supposed to be in this picture. You have so many normal flora. And some bacteria can be both normal flora and pathogen. Okay, so in the old concept, we believe normal flora is one type, pathogen is another type. When you get infected with this bacteria, you always get sick. But now we know, like, not always true. Sometimes the pathogen live in our body, but get some benefit to our body. But if amount of the bacteria increase over than normal amount, that will harm us. Okay, for example, Clostridium difficile. Clostridium difficile is an aerobic bacteria live in the colon. Okay, normally we have amount of Clostridium difficile, but if some people take broad spectrum and then the drug kill most of bacteria, but not kill this Clostridium, what will happen? Clostridium will overgrow because when they live together, the other bacteria kind of control each other to make the population is in balance. So once they grow a lot, it causes disease, pseudomembranous colitis. You get the inflammation on the membrane of colon, or candidiasis caused by a yeast, Candida albicans, in your mouth, in vagina, something like that. This is the causative agent of vaginosis. In Thai, we call ตกขาว. So when we know antibiotic can kill bacteria, can stop bacteria, can have spectrum, okay? we probably need to know the route of administration of antibiotic. Normally, we use 3 routes. Oral is mean the antibiotic in capsule and you take it. Intramuscular, you just inject it in the muscle, okay? Intravenous, you need to dip antibiotic into your vein, okay? 3 major routes. If you take intravenous, antibiotic go to your blood and concentration is very high and very effective. Sometimes, I don't know, this is not a good idea. If you get sick and you need to cure the very quick, then you go to see doctor. Doctor asks you stay in hospital for 4 or 5 hours and then put the intravenous and put antibiotic inside that. You get IV. Once you sleep and wake up, you feel much better because antibiotic go directly to the vein and then move to your heart and pump out to everywhere of your body. But if you inject it in muscle normally doctor will inject it here or your arms, shoulder, or your bum. Then you spend some time until the antibiotic diffuse to the site. Okay, but the oral is also slower, a little bit slower. Oral and intramuscular may be similar. Well, when we classify antibiotic by structure, I don't want you to remember structure, but I want you to understand the basic core structure. There are a lot more than this, okay? This is 6 group that is very, you probably see here or read through the text very often. The first group is beta-lactam ring. Here, beta-lactam. Beta-lactam is mean this the red structure. Structure of cyclic with 3 carbon. One nitrogen, double bond O. This is beta-lactam. The side ring can be 5 member or 6 member, that's okay. Second one is tetracycline. Tetra is means 4. That means 4 cyclic linked together. Okay, that's structure of tetracycline. The class of antibiotic can be so many antibiotics, but they all share the same structure in the core structure. Okay, you probably see this structure is carbohydrate, sugar. This is sugar and another sugar, and you see amino group NH2. This is amino group, amino group, amino group, amino group. So many amino group. So this structure called aminoglycoside. Okay, aminoglycoside. This structure, you see the big member ring. Okay, 13, 14 member ring. I can't remember. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 13 member ring. This is macrolide ring. So macrolide can link with sugar or non-sugar, that's okay. Or maybe a bigger structure. But this is macrolide ring. Okay. 2 of the carbon 6 carbon member ring here, plus 1 fluorine called fluoroquinolone. Okay, this is another group. And sulfur with 2 double bond O here is sulfonamide. There are so many groups more than this. This is the classification of antibiotic by chemical structure. When we say this bacteria, you hear a lot more in the other lecture. Treatment using beta-lactam drug, that means any drug that has this structure. But if we classify antibiotic by the mechanism of action, the mode of action, we classify them into 5 different groups, okay? The first one, antibiotic that inhibits cell wall synthesis. Cell wall synthesis is we are now talking about every chemical that inhibit peptidoglycan biosynthesis. Okay. For example, penicillin, cephalosporin, carbapenem, vancomycin, something like that. The second group is inhibit membrane function. Membrane function, so basically biphospholipid of bacteria and human are quite similar, okay? But some protein on membrane, probably different from human and bacteria, right? So the drug point to that target, not just membrane bilayer. Some drug inhibit protein synthesis, so stop bacteria to produce protein, okay? Inhibit nucleic acid synthesis, so they block bacteria to produce nucleic acid, to replication, to transcription, and inhibit metabolism of bacteria, okay? I'll say the drug that inhibit bacteria with the same structure or same pathway or same mechanism that host also has, that might be not a good idea, okay? So in this case, 5 of these are quite well, I would say quite effective, but in different level. The drug inhibits cell wall biosynthesis is very good because humans don't have cell wall. You can dump a lot of drug with not very high toxicity to human. Okay, the protein synthesis is another one that is a good target because protein synthesized by bacteria and hosts use different type of ribosome. Yesterday we learned already, right? Bacterial ribosome and eukaryotic ribosome has different 78 and 88, right? So they target differently. That's perfect. Nucleic acid biosynthesis also a good target because DNA polymerase of eukaryote and prokaryote are quite different. So metabolism, this is also good, but not easy to find the pathway that present only in bacteria, not in human. So plasma membrane function, we need to point to some protein that present in bacteria, not in human. Membrane protein. So there are 5 basic mechanisms. Let's start from cell wall, inhibit cell wall. The antibiotic that inhibits cell wall, majority is supposed to be beta-lactam ring, okay? Beta-lactam ring can, well, when we talk about this, we mention the group of penicillin, cephalosporin, monobactam, carbapenem. I show you 4 of these, not just for you to remember structure, but you see they are all share the same core, okay? Beta-lactam probably linked with 5-member ring, or some beta-lactam linked with non-member ring, that's okay. 6-member ring, different class, but they all function in this site. This is the functional structure, okay? Let's see, penicillin. Penicillin is the first antibiotic we know. Previously, the first antibiotic we know is penicillin G. The pen G, you see, this is beta-lactam. And then once we apply pen G to bacteria, they kill bacteria very good, but bacteria fight back by develop some resistant mechanism. Bacteria develop an enzyme to digest penicillin. Okay, then we just develop the the side chain of penicillin here by adding an ether group here. Okay, then pen G is more, they actually change the property. The pen G normally get injection, and pen V you can take by oral. Both of them can be digested by penicillinase of bacteria. Okay, once we know that, we try to modify the side chain to be the new generation, oxacillin and ampicillin. Both of these resist to beta-lactamase enzyme of bacteria. So once you take the antibiotic, so once you take antibiotic, and then bacteria cannot digest the antibiotic so quick. Okay, let's see the structure of.
Ajahn ka. I'm sorry for another interruption, but there's like another student trying to enter the class.
I accept him already.
Oh, thank you, my apologies.
Sorry, Ajahn.
So try to perform, because I need to send the, I cannot, the MUIC asked me to download the form that if you perform to the class early or late, because the Zoom record everything. They asked me to send that, and this is reflect to your performance. Okay, so next time come online on time.
Yes, Ajahn. Thank you so much.
So this is the peptidoglycan biosynthesis in brief version. Start from, they have metabolite of a sugar, okay, and acetylmuramic acid, and then add different types of amino acid to the muramic acid. And then this is the cytosol in the cytoplasm. And then another type of sugar, and acetylglucosamine, come to link with the complex one to get the subunit. And these subunit need to move from inside of the cells across membrane, go to the, if gram-negative, go to periplasmic space, if gram-positive, go to somewhere around the cells, okay? And then this subunit need to link more and more and more, polymerize to be a very long polymer, okay? And they use another enzyme called transpeptidase, link the amino, side amino link together, okay? This enzyme, transpeptidase, sometimes we call penicillin binding protein, because penicillin can come across the porine protein. Still remember? When penicillin come to the periplasmic space, they bind to the penicillin binding protein, or PBP. And once the drug bind to the enzyme is inhibit. Once enzyme is inhibit, they cannot make a cross-link of peptidoglycan. Peptidoglycan without cross-link is loosely, and it's not rigid and not provide a strong to the cells, particularly gram-positive cells that need a strong peptidoglycan, because gram-positive cell allow osmosis inside the cell and get high pressure. If cell wall is loosely, the cell might be burst or lysis because of the over osmotic pressure. That's why penicillin kills bacteria. Okay, so everyone can follow this. Bacteria develop enzyme called penicillinase to digest the beta-lactam ring. Okay. Once the bacteria digest beta-lactam, well, the penicillin will be penicilloic acid, so lose beta-lactam structure, and this cannot allow the medicine bind to PDP. Okay, so the way that scientists can do keep changing the R group. Well, the new R group here make the enzyme cannot recognize, but enzyme bacteria keep mutation to recognize the drug all the time. Okay, this is the war of human and bacteria. So beta-lactam come to the polyplasm and bind to PDP and then block bacteria. Then bacteria use penicillinase, cleave the beta-lactam, then it cannot bind to PDP. Bacteria die, and bacteria keep develop the enzyme penicillinase until we use methicillin. Okay, methicillin is the last. Methicillin is special one. It designed for tolerate penicillinase enzyme. We call penicillin resistant. So bacteria that produce penicillinase called penicillin resistant because they tolerate penicillin. Methicillin is penicillin resistant antibiotic or penicillin resistant penicillin. That means it is penicillin to fight with the resistant bacteria. Okay, and penicillin is work well. Methicillin is work well bind to PDP until bacteria mutate to change PBP itself, not penicillinase. Change PBP that make the active site change. Methicillin cannot bind to PBP, but this is very serious. We call bacteria, particularly Staphylococcus aureus, or called MRSA, methicillin-resistant Staphylococcus aureus. Once bacteria mutate to PBP like this, that means all class of penicillin, all antibiotic cannot work. So these MRSA tolerate all penicillin immediately because they ignore active site of penicillin. All right. So the way we treat with penicillin is we need to target by the other antibiotic. It is vancomycin. Vancomycin is the last drug to fight with Staph aureus. Okay, they block in different points instead of cross-link. They block the translocation of subunit across membrane. So that is the tactic. This is the last weapon of human against Staph aureus. Okay. This is another group of penicillin called cephalosporin. Cephalosporin also be a beta-lactam, but you see different side chain, okay, different side ring. So this also resists to penicillinase. Okay, that's the thing. Another class of antibiotic is inhibit protein synthesis. Because the protein synthesis, because they go to point at large subunit, okay? Tetracycline, so they block the tRNA go to bind the inside the go to bind the anticodon. Chloramphenicol also block peptide bond formation. Okay. Aminoglycoside, this is another group. Okay, disrupt the reading of mRNA. So these are point of the protein synthesis antibiotic. Antibiotic inhibit nucleic acid biosynthesis. Could be rifamycin, the one that you probably remember, but don't worry, I will mention you again next week. Rifampicin. Rifampicin can penetrate to the host cell very well. That means when you take this antibiotic, rifampicin will pass to your cell and action inside your cell. That's good for intracellular bacteria. There's some bacteria when get infected in your body that go to escape immune response by hiding itself inside your cell. So rifampicin is one that go to kill bacteria inside your cell. That's very good. One of those bacteria are mycobacteria, positive agent of tuberculosis. That is the one. Quinolone, still remember, two 6-member ring with fluorine. For example, ciprofloxacin inhibit DNA gyrase. That means inhibit DNA replication. DNA gyrase in bacteria in eukaryote are different, okay? Polymyxin B inhibit membrane function, okay? This, well, the medicine that inhibit membrane function is not so many, okay? Sulfonamide inhibit metabolism. Okay, that's the idea. So bacteria need to fly back to the overuse or abuse antibiotic. Okay, we call antibiotic resistant instability that bacteria fly it back. Okay, we concern a lot about using antibiotic probably kill the, if you see this, the yellow dot mean general bacteria in your body, but the red dot is bad guy, okay? It resistant bacteria when you apply antibiotic, but sometimes coincidentally mutate and then resist to the antibiotic. When most of bacteria die, but this one can get benefit from mutation and tolerate antibiotic, they start increase in population until the population of bacteria in your body change from general yellow one to the resistant strain, the red one. So then you suffer with the antibiotic resistant bacterial infection. That's the point. Or some of your friends asked me by email, if you guys need to take antibiotic and unfortunately you get allergy from antibiotic, probably from hypersensitivity type 1 or maybe type 4 hypersensitivity by the using of antibiotic, should you continue antibiotic to the final day to protect the antibiotic resistant, or should you stop taking antibiotic? The answer for your friend is stop taking antibiotic immediately because allergy is more serious than the resistant strain generation. But you probably need to go to see doctor to change the antibiotic. The reason when patients get allergy for antibiotic because they probably allergic by side chain or the core structure of antibiotic. Doctor can change another group of antibiotic because there are so many antibiotic in use of the same bacteria. Okay, So then once doctor change to another group, for example, if you get allergic for beta-lactam, penicillin cannot use with you, then doctor probably change to aminoglycoside. That's action with cell wall synthesis also is work.
อาจารย์
Yes.
so I heard that penicillin allergies are really common for some reason, especially IgE allergies. Do you know why?
Well, it is related to genetic of host also. So the allergic is of any medicines, we refer to the term pharmacogenomic. So now we sequence human genome, okay, from a group of patients who get allergic with medicine and non-allergic with medicine, and we compare DNA of allergic group and non-allergic group, and we found some gene. Mostly we point to HLA gene, okay, because HLA related to the presentation of antigen. Once you get some HLA, you potentially prone to allergy, something like that. When doctor know that, in the future, doctor will change the way to use that antibiotic to another group that your HLA cannot present, or you don't have a marker to show that you're prone to allergy. The thing like this we call personalized medicine. The medicine that related to individual. In the future, doctor probably check your genome before they give you a medicine, because the same medicine sometimes give with patient A is very well, give with patient B is not quite well, because the ability of patient B digest a lot of medicine, the concentration of medicine in patient B lower than A, then patient B probably need double dose of medicine, something like that. So every medicine will precise to individual, something like that.
Okay, thank you.
The antibiotic resistant. Why we care of antibiotic resistance? Because they can lead to treatment failure. Okay, if we, well, you probably cannot imagine. I suppose all of you probably younger than 23 years old. About less than 100 years, Thailand and everywhere in the world meet the situation of no antibiotic. So when we have no antibiotic, you need to imagine you probably die from so simple disease. For example, common cold. When you get common cold, or you get abscess or pimple, the abscess เป็นฝี you prone to die by abscess, the easy abscess. But nowadays, you take antibiotic, abscess is so easy to kill. Okay, so we are almost meet the situation of no antibiotic use, because bacteria trying to resist almost all antibiotic use nowadays. So that is the problem. Why we need to face the big problem. Ability to modern medical procedure, when doctor want to operate, want to do something with to treat you by surgery, you get wound. And once you get wound, you're prone to get infection very easy. Okay, so antibiotic is overused after you get operation. Antibiotic impose a major economic burden. That's of course, just like coronavirus now. Any country stock antibiotic, okay? And cost many deaths around the world, even in Thailand, cost quite high. More than 38,000 people a year dead by antibiotic resistance. It means no antibiotic, no method to kill those patients because they get infected with bacteria with no antibiotic. Okay. But unfortunately, people or country that meet the situation of antibiotic resistance are poor country, not a very rich country. We probably see the causative date of antibiotic resistance in Thailand, in Africa, in India, in China, but quite less in Europe, in the US, something like that, because of sanitation. Well, I'll say bacterial infection will cause with poor country. The rich country will get problem with viral infection. Okay, so we don't so sure like applying antibiotic can cause bacteria resistance, or actually bacteria resistant because they gain the virulence inside the bacteria itself. We don't know which one is correct, but the fact that antibiotic can be a selective pressure to force bacteria, and just only resistant strain can pass to the pressure. That's the fact. Okay. Mechanism to resist an antibiotic can be intrinsic, natural, or acquired. Acquired means genetic method. Okay. Let's say intrinsic resistance. For example, penicillin attacks cell wall biosynthesis. Some bacteria do not have cell wall. They evolve itself to have less cell wall. For example, mycoplasma, no cell wall. Okay, that's tolerate automatically, tolerate the drug automatically. Or they generate the efflux pump. Okay, efflux pump is type of membrane protein to pump drug outside of the cells. Even drug go to the bacterial cells quite a lot, but the rate of pump of antibiotic out is quicker than the effect of the drug. That's okay for bacteria. Or sometimes bacteria has some enzyme to inactivation of antibiotic. Mm-hmm. For example, cephalosporinase in Klebsiella makes cephalosporin is not work in Klebsiella. That's intrinsic, but it acquired resistance is something happened by genetic. The genetic, we term the antibiotic resistant as R gene. R gene is mean resistant gene, whatever, ampicillin resistant, tetracycline resistant, whatever we call R gene. The R gene from bacteria can transfer to another bacteria by conjugation, by transformation and transduction. Conjugation need pili, 6 pili, sent from one cell to another cells, right? Transformation happen by bacteria that host the gene has been dyed and lysis. When the cell lysis, some part of DNA broken, DNA probably uptake into another cells by transformation, and then if those bacteria, those DNA that go inside the cells has some homologous, they can get recombination and transfer into the chromosome. Transduction use virus, in this case, I mean bacteriophage, infect with the bacteria with R gene and incorporate R gene in viral genome. Once the virus lyse bacteria. They get infect another bacteria, then they transfer our gene to the second bacteria. Okay, our gene can be in plasmid and spread within bacteria. I mean, spread from plasmid to the chromosome. Well, if the R gene present on plasmid, they potentially lost plasmid, and bacteria that resist to antibiotic can reverse back to the normal bacteria. Okay, but if they persist in the nucleus, sorry, not nucleus, the chromosome, so this characteristic will be kind of permanent. So transposons are a major role to transport the gene from plasmid to chromosome. Biochemical mechanism of antibiotic resistance can be prevent the drug accumulation, where there's so many techniques. Quorum sensing. Quorum sensing, I mean, the number of bacteria. When you get infected with one bacteria will secrete some protein or excrete some small compound. When bacteria keep multiplying, the number of compound protein that they secrete or excrete out is more intense, more condensed. And then that is tricked to the other bacteria to let them resist or do something. That is a signal among bacterial population we call quorum sensing. Okay. Enzyme activation, protein target site, modify pathway, that is the mechanism that bacteria try to tolerate antibiotic. This is the mutation, one of these mechanisms, okay? Normally, this is outer membrane of gram negative, okay? Antibiotic pass through the polyplasmic space by porine protein, okay? The way that, an action by peptidoglycan, the way bacteria mutation to loss porine protein, then antibiotic cannot go inside the cells. Or reduce number of porine protein. Normally they have a lot. So once number of porine decrease, the amount of antibiotic that can go inside the polyplasm less than it should be, and change structure of pollen to make the channels narrower, then antibiotic cannot pass through the channel. That's another mutation, okay? Or efflux pump triangle. The black triangle is mean antibiotic. Here, outside the cells, concentration. Well, antibiotic move inside the cells, concentration should be accumulate a lot. When they pass through, for example, tetracycline use this efflux pump to protect themselves. Tetracycline move inside the cell and pass through membrane. And then the pump take tetracycline out all the time. This is the way that bacteria spend mechanism to use. This one also, I also tell you, penicillin bind to PDP, mutation to change the binding site. When they change the binding site, this is mechanism that protect target site, okay? Then antibiotic cannot bind to the target, then bacteria survive. Okay. So before we stop this lecture, I just add some slides about laboratory method for antimicrobial susceptibility testing. Okay. We need to test antibiotic susceptibility for it is very useful for some bacteria, such as Staphylococcus aureus. When doctor diet, like this patient get infected with Staph aureus, knowing the antibiotic resistance is very useful for treatment, okay? Because doctor want to know penicillin can use or not, or vancomycin can use or not. So media for testing, we use Muller-Hinton medium. If you use broad, it is Muller-Hinton broad. If you use agar, you use Muller-Hinton agar. And this is the normal color of Muller-Hinton. The reason we use Muller-Hinton because actually it's more or less like nutrient agar plus starch. They have starch inside that, and starch can absorb all toxic from bacteria that bacteria produce, okay? Some bacteria produce a lot of toxic, then starch inside Muller-Hinton absorb all toxic. So bacteria will die by the effect of antibiotic alone, not antibiotic plus toxic. So Muller-Hinton help you measure ability of antibiotic very exact, okay? So conventional, there are so many techniques to test the susceptibility. One that is very easy, and I want you to know the idea is make a dilution of antibiotic, and you test with one bacteria. For example, if I want to test Staph aureus with one antibiotic, then I dilute antibiotic from 4 microgram per ml, make serial dilution. Half dilution is 2 and 1, 0.5, 0.25, 0.125 microgram per ml, okay? And apply the same amount of Staph aureus. For some concentration of antibiotic, Staph aureus cannot grow. So we want to know MIC. MIC is minimum inhibitory concentration of the drug. The concentration, that means the concentration that the minimum concentration that still show up clear or bacteria cannot grow. In this case, MIC equivalent to 1 microgram per microliter, because 0.5 you still see some turbid inside. If you want to read that, you probably shake the tube. If you not see any turbidity, then you read that concentration as MIC. That's minimum inhibitory concentration. We can know MIC for the efficiency of the drug. But inhibit, probably we refer to bacteriostatic, not bactericidal, right? Then, well, the first day you culture bacteria. Second day, you just apply to MIC. Third day, you just use one loop, streak on the plate, pick up the solution on the clear tube and streak to the new plate, okay? In this case, bacteria in the tube 1 microgram per ml can grow. That means this concentration has efficiency for stop growing, but not efficiency to kill bacteria, okay? But the minimum bactericidal concentration is 2 microgram per ml. This is the concentration that clean the clear bacteria. So we probably talk about MIC and MBC. You need to know how different between two of these. So when another technique is easier, you just spread Staph aureus or bacteria you want to know, and put the paper disc with antibiotic on the smear of the spread of bacteria and incubate overnight, then bacteria will grow and antibiotic will diffuse in the agar and inhibit bacteria. The size, then you probably measure the size of clear zone, okay? The way to measure the size of clear zone, you need to measure across the disc two-way and calculate the average, okay? We start from 6 millimeters, the curve, because the disc itself has 6 millimeter diameter. If you observe nothing, that means 6 millimeter, okay? So the size of bacteria that is show up with very narrow clear zone, very small clear zone here. This is the resistant. And then the bigger size of clear zone established susceptibility zone. In between, we refer to intermediate, just to know the technique. The interpretation is the job of doctor. The last one is much better, is E-test strip. E-test strip, you just smear bacteria and apply E-test. E-test is a strip that they immense antibiotic inside the E-test already. And the concentration of antibiotic will gradient on the paper. They have scale. You just put the strip on in this area. The concentration of antibiotic is quite a lot. Then they diffuse in the zone. And this is smaller, the zone is narrower until the area that you can read by spread of bacteria that touch to the strip. So that's E-test. The good point of E-test is you can read MIC quite precise. But if you use the tube, you don't know actually MIC is 1 or 0.9 or 0.8 until 0.6. Okay, so kind of big jump. The value is not precise. This is the point. Oh, that's all about the antibiotic.